Hello everyone, welcome back to another session of RNA biology. So we were here in the previous class that we were discussing about the role played by a protein YY1, that is also called as yin yang 1. This protein helps in tethering the Xist onto the chromosome, the place where it is being produced. So if you use YY1 siRNA to block its translation, then you will not have the ability of this Xist localized onto the corresponding chromosome. So that is, and also the methylation status H3K27 trimethylation, which is a post tethering or post Xist attachment onto the X chromosome. Then only you get this H3K27 trimethylation markers, so that will go missing. So the take home message from these experiments is that Xist will be able to attach to its target, that is the X inactivation center, with the help of a protein called YY1. So how is the mechanism of tethering or attaching? So it works something like this. So you have this Xist RNA, and this is the place where it is supposed to be pairing the nucleation center, the X inactivation center, and the protein YY1 is right in here in the middle. So somewhat like a glue, like a Fevikwik or Fevicol glue that is attaching onto it, and once it is attached, then that is triggering the further events such as PRC2 recruitment for the H3K27 trimethylation, etc. So the YY1 binding occurs onto the Xist RNA through a specific domain called YY1 binding domain that is present on the Xist RNA. So Xist RNA will be able to bind with the YY1 protein, and YY1 protein in turn have an affinity for binding onto the X inactivation center, and you can see the PRC2 complex also have a location on the Xist RNA. So this brings in the PRC2 complex. So to recap this whole thing, this is the X chromosome, this is the X inactivation center, and from the X inactivation center you have the production of Xist RNA, and Xist RNA have got YY1 protein binding site, and YY1 protein is able to bring the Xist RNA onto the X inactivation center, that is the nucleation center, and this Xist in turn has got binding sites for PRC2 complex, polychrome repressor complex 2, which is required for the H3K27 trimethylation. So this is the whole process. Those who are interested to know more about the importance pertaining to the X inactivation center can read this article that is published in Cell few years ago, and this paper discusses one more important thing, that is the mutation rate is seen to be very high in the inactive X chromosome, like this so-called bar body, which is meant to be not functional, is accumulating lots of mutations compared to the counterpart, that is the X active or active X chromosome have got less mutations, or it is just like any other normal chromosome, whereas this inactive X chromosome accumulates lots of mutations, and we call it as hypermutations. Let us see with some more detailed observations. So the X inactive means non-functional X chromosome in females accumulates somatic mutations much more during the tumorigenesis. Let us see a normal cell and a cancer cell. Okay, so in a female, if you have cancer, those cells also have got two X chromosome, one is active, one is inactive. So we are comparing the inactive and active, and how does it differ. So in this graph you have somatic mutation. What is somatic mutations? Mutations that is occurring in a housekeeping gene or a gene that is responsible for the normal day-to-day functioning of the organism. So that is what we refer to them as somatic mutations. And the other genes are meant for determining the sex of the organism. So they are not called somatic genes, they are sex specific genes. So the somatic genes can be present in the X chromosomes also, and the somatic mutations are often leading to some serious trouble in the organism's survival and other day-to-day affairs. So what you have to see here is the number of mutations. Bottommost of this y-axis is low, topmost of this y-axis is high. That means low number of mutations means the graph will be restricting towards the bottom, whereas large number of mutations means it will be pretty high. So the somatic mutations across the chromosome, starting from chromosome 1 until 20, 21, 22, and this is the 23rd in human, that is the X chromosome. So you can see here in X chromosome there is more of the mutation. That means there is the X chromosome is shown in this blue color. So what happens, the same female cell, same means not the same individual, another female cell that has got mutation rate in somatic chromosome or somatic genes and compared to the X chromosome. So you can see here, compared to a normal healthy cell, the number of mutations are pretty low, and you can see throughout the chromosome it is almost double. You have some way or the other mutations because it is not a normal cell, it is a cancer cell. But what you have to see is when all the somatic chromosome or somatic genes have got almost double, whereas the X chromosome, it is almost four times more mutations. So when somatic chromosome have got 2 times mutations compared to a healthy normal cell, the cancer cell have got almost double compared to a normal cell, but the same cancer cell, the inactive X chromosome usually will accumulate a plenty of the mutations, almost 4 times more. So in cancer, X has up to 4 times more mutations than in autosomes. And one more thing, if you see in X chromosome, which is inactive and active, if you compare, the inactive one has accumulated more number of mutations compared to the active one. So this is proven fact in across a bunch of cancers. So it's an overview of 402 cancer genomes analyzed for this study in that paper, that Cell paper. So you can see, I will not go through each and every cancer, that is acute myeloid leukemia, B-cell lymphoma, breast cancer, so and so. So you can see the number of females studied and number of mutations studied or analyzed, and several other related publications are also cited here. So what we understand that in human female, the mutation rate is much high in the inactive X chromosome of a cancerous cell or cancerous phenotype. So that is what this study has proven. So the inactive X chromosome in female accumulates somatic mutations during the tumorigenesis. We kind of know the reason because we know because of the high level of compaction of this chromosome, it is taking lot of time to unpack it, and because of this, towards the end of S phase is the synthetic phase, that is the phase where the DNA replication takes place. That is the phase where there is any error in the DNA replication that has to be monitored. Everything happens in that phase. However, if this fails, if it could not do, every chromosome is vulnerable for changes. The so-called proofreading activity helps a lot in fixing the errors. But what happens, since the inactive X chromosome takes a lot of time to unpack it, it do not get enough time for repairing the possible damages. So if there is a damage, that will be maintained as it is. There is no time to repair it. It will move on to the G2 phase and then M phase. But it is not a deleterious thing in inactive X chromosome because none of these genes are expressed. Hence, there is no one to take care. There is no mechanism to take care of fixing the mutations in the inactive X chromosome. So the rare mutations is possibly not different from autosomes. The X inactive or inactive X chromosome is not repaired due to its late replication, because in order to repair you need to have enzymes available in the cell, because since it is almost towards the end of the S phase, the enzymes required for repair are missing. But soon after the replication is done, the X chromosome is made inactive immediately. So there is no scope for repair, and hence it prevail the mutations as it is. So now let us see how the genetic imprinting is undertaken by shRNA. So we know to some extent that what is genetic imprinting. I gave some examples in the previous class also. Imprinting basically means you are designed, you are guided in a particular manner. That means you have to do so and so. Like from childhood you have been given some specific mannerisms or manners you have been taught, and you follow accordingly. You say in school, you say you have to wish good morning to your teacher, and like that teacher, wherever you see in any time later in your life, you will still learn to wish. So it's almost like a imprinted behavior or imprinted character, and no matter what, you will stick with it. So many times many genes also follow an imprinted behavior when it comes in terms of expression. So this non-coding RNAs contribute to a large extent in controlling the imprinted characteristics. So genetic imprinting is a process which results in the expression of only one allele of a gene, and that is called monoallelic gene expression, while the allele originating from the the other parent is silenced. We have to understand you have two copies of each gene and we call it as allele. Say gene A, you have two copies of it. One came from father, one came from mother. So we call them as a paternal copy and maternal copy. These are alleles. For many genes, the copy that came from father will be allowed to express, and for many genes, the copy that came from mother will be allowed to express. In some cases, both the copies are expressed, and we call them as biallelic gene expression. When only one is allowed to express, that is called monoallelic gene expression. But this monoallelic gene expression to a large extent is imprinted. Imprinted means it can't happen that for a given gene, okay, like X chromosome we saw that randomly one of the X chromosomes inactive much later stage in the embryonic development. Earlier it is the paternal copy that is made inactive, X chromosome, but when it comes to imprinting, it cannot be random. Some genes, many examples we will see them. Some genes, a given gene, it will be expressed if it is coming from the father, whereas for some other gene it will be expressed if it is coming from mother. So what can be outcome? Let us think this way. Say paternal copy had got some issue, it is not correct, it is having some mutations. The maternal copy is perfectly fine. So you have an option of expressing the good copy, but it will not happen because this gene is following imprinting rule. So if the paternal copy is supposed to be expressing for that particular gene, if that paternal copy is having some defect, then you suffer, you have problem. Although you have a perfectly okay maternal copy with you, that copy is not allowed to express. So this is the importance and beauty of the imprinting. So many diseases occur in a person because of this imprinting. Imprinting do not cause the disease because of the condition like I told you, because that gene is supposed to express only from father, or so and so gene is expressed only from mother. If the father or mother gave you a mutated copy, then there will be a problem in terms of its functionality, or based on how severe the phenotype is, the organism suffers. So imprinting is not just a way of living, sometimes it can cause really deleterious effect. The process is somewhat similar to dosage compensation. Imprinting, we studied imprinting to some extent while discussing the dosage compensation also. The differences of expression from both the alleles are due to different states of the chromatin. Chromatin we know can be of two types, euchromatin and heterochromatin. Euchromatin means loose chromatin and DNA is accessible for transcription, whereas heterochromatin means it is tightly packed chromatin where the gene expression is poor or nil. So the chromatin is not opening up, DNA is not accessible, so that's called heterochromatin. So the differences in expression from both the alleles are due to different states of the chromatin and is also having differential methylation of the DNA itself. We have seen S3K27, histone 3, lysine 27 trimethylation in the case of X inactivation, and many a times it is associated with repressive histone modification, that is S3K27 methylation, whereas S3K27 acetylation is an indicator of gene expression, active gene expression. So we should understand that the differences in the gene expression events to a large extent goes to the imprinted status of a gene, and not all genes are imprinted. A fraction of the total genes are imprinted. Activity of small heterochromatic RNA, we call them as shRNAs, appear to be important for establishing and maintaining the imprinted status of the gene. So like X chromosome, we have seen it, making an X chromosome inactive is the first part only, but you have to keep maintaining. If you fail to maintain it, then that can lead to trouble because dosage compensation will be imbalanced. Some situation similar to that of Down syndrome can come. Not exactly the same thing, like Down syndrome is happening mainly because of lack of such dosage compensation. You have 3 copies of chromosome number 21. So something similar can happen if there is a leaky expression of X chromosome. So same problem can happen if there is a leaky expression of one of the imprinted genes. Say a paternal copy has to be turned off, you don't want that gene to be turned on. If you turn it on due to whatsoever problems, then that can lead to an imbalance in the dosage compensation. So what we should understand, genomic imprinting is a way of ensuring dosage compensation, and it's also a way of ensuring heterozygosity. That means heterozygosity here I am referring to as the outbreeding. Like many species, like you may have heard about like some reptiles, like you may have heard about this Komodo dragon, which is seen in the Komodo Islands of Indonesia. So this is one of the largest reptile, like that is almost the size of crocodile or even bigger than that, but land dwelling mainly. So that's I think around 6 to 7 feet. You can read about it. So this Komodo dragon, they exhibit a feature called parthenogenesis. Many lizards exhibit this feature called parthenogenesis. What is parthenogenesis? The female can lay viable eggs without the help of a male. It do not need fertilized egg. Like if you have seen many chicken farms, the female chick will lay eggs without mating with a male. Without fertilizing, it can lay eggs, but that egg will not hatch. Usually it will not hatch. It is okay for eating, etc., but you cannot hatch it. For hatching, it has to be a fertilized egg. But whereas Komodo dragon, if they are laying eggs, it can hatch, and this will give rise to both males and females. Eggs are laid by only female, males will not lay eggs, but the hatched ones, they can be males and females, because many of these reptiles like crocodile or some turtles, they follow temperature dependent sex determination. So when they lay eggs, they will lay vertically in a pit. So in summer season, since the eggs are put in a vertical pit, then they will face a gradient of temperature. So usually in the case of crocodile, it is between 31 and 32 or 30 or 32, I have to check it, but around that range. Between 30 and 32, I think 30.5 to 31.5, something like the very narrow range, you will get males. Above 31.5, all females. Below 30.5 or 29, something around that range, around 30s, plus or minus 1 degree, you will have males. Above and below, you will have females. So this is called temperature dependent sex determination. Similarly, Komodo dragon also, although the female is laying eggs, that is viable eggs, that is hatchable eggs, that will give both males and females. So this is an advantage for Komodo dragon because it can propagate itself, because otherwise chances of finding a male in the same island is less. So a female landed in an island, if it could not find a male, then chances of propagation is less. Hence it become advantageous. Parthenogenesis become advantageous, and now you have both males and females from this offspring, and they will follow a sexual breeding. Why I said this? Because this imprinting ensures nature wants outbreeding. Nature do not want parthenogenesis. Nature want outbreeding, mainly because you should be able to incorporate diversity, more and more varieties, so that the survival of the fittest, as per the Darwin's theory of evolution, survival of the fittest can be ensured. How can you ensure the fittest population being formed in a society, in a population? Because you have to bring in changes, not from other species, from the same species itself. You need to bring in changes, you need to bring in variety. So to ensure this, you need to indulge in the sexual breeding. So the imprinting is a way of ensuring that sexual breeding is assured. Say for example, let us think a female that is laying eggs, and of course it has to become diploid, etc., by retention of the first polar body or second polar body. That's a different thing. Let us don't go into the details of it. But if a organism is following imprinting, a female gene is not going to be expressed in the next generation. Only a male copy is supposed to be expressed. Then parthenogenesis will fall apart. That is why many mammalian species or many bird species, like I told you, chicken is laying eggs. Why it is not able to give rise to a hatchling? Mainly because it has many genes that are imprinted and it will not be able to express in the offspring. So imprinting makes sure that the organism has to make a copy of the new organism only through the sexual breeding, that is male and female mating. So activity of various this heterochromatic RNAs, shRNA, is not limited only to genetic imprinting. So they have many other roles, but shRNA plays a major role in the initiation and maintaining of this imprinted status. No leakiness is allowed. So genomic imprinting is a feature that has to be strictly followed and effectively followed in various organisms, especially in birds and mammals. So you can read more about this heterochromatic small RNAs in this review, that is RNA mediated epigenetic regulation of gene expression. It's a quite interesting article, and since it's a review, you will be able to understand it much better. So DNA and RNA recognition models of shRNA initiated chromatin condensation. Means in this section we will try to understand how a shRNA is contributing to the maintenance of this imprinted status. So we have two models. One is a DNA recognition model and an RNA recognition model. We know all genes are located in the DNA and RNA is formed from the specific loci or location in the DNA. So you have this DNA and you have this DNA recognition model, and this shRNA comes into picture. shRNA has come from a given location of, it has its own gene, and this shRNA being small, it can find a specific sequence, like just like how miRNA binding onto the UTR of a given gene, and it will cause its RNA induced silencing complex, and it will cause the repression of those genes. So like that, this shRNA can find one of the DNA strands and it will pair complementary, it will have match with the complementarity. The other DNA strand is lying empty. You may remember that R loop hybridization, how introns were discovered, something similar. It will go and pair. Now, once this pairing has happened, it can bring in other protein factors such as silencing factors and also histone modifying enzymes. Once they have occupied, that will spread across the neighborhood until there is a boundary is reached. You have seen similar thing happening to the entire X chromosome. Here, entire X chromosome is not affected, but a part is affected. And in the RNA recognition model, similar thing is happening, but instead of binding onto the DNA, it is binding onto the newly formed RNA, which is not completed. It's a nascent transcript, smaller, longer and longer. And once this region is exposed, this shRNA will come and pair onto the newly formed RNA, and it will bring again the silencing factor and histone modifying enzymes and cause the spreading of this silencing events across the neighborhood, not the entire chromosome. It is only in the neighborhood, and make sure that this gene is never expressed. So we will see more in detail about this mechanism of genomic imprinting and more important way of gene regulations in the next class. Thank you.